Ngalakin Khadijan Nyunga Boja.
This is the second lecture in the lecture series on oxidative stress. In the first lecture, I introduced the concept of reactive oxygen species, talked a little bit about the terminology, gave you some examples of the major classes of reactive oxygen species, and discussed some of their properties. I then moved on and talked about some of the sources of reactive oxygen species, and our interest was in the endogenous sources of reactive oxygen species, and then I discussed some of the challenges in actually measuring reactive oxygen species. What I would like to do in this particular lecture is talk about one of the major ways in which reactive oxygen species can affect cell function, and that is via oxidative damage. And this has certainly been a very central focus of reactive oxygen species and oxidative stress research in general over the last several decades. And so for this particular lecture, we'll look at how reactive oxygen species damage proteins, lipids, and DNA. I'll discuss how some of the products from that damage can be used as biomarkers of oxidative stress, and we'll also highlight some of the limitations of some of the assays associated with these approaches. In discussing how reactive oxygen species cause oxidative damage, most of our focus is going to be on hydroxyradicals. And the reason for this is that hydroxyradicals are probably the major way in which most oxidative damage is manifested. It doesn't mean that other reactive oxygen species cannot cause damage, such as superoxide, for example, but it is thought that hydroxyradicals are the major causes. Now, hydroxyradicals can basically attack any cellular component. So, for example, they can attack the lipids in membranes, both of the cell and the mitochondria and nucleus membranes, as well as endoplasmic reticulum membranes. This can lead to lipid peroxidation. You can also have damage of proteins in the membranes as well. And if it's sufficiently severe, then you can have leakage of molecules such as calcium into the cell, sodium in the cell, and that can end up causing cell death by necrosis. Other targets are proteins, so we can have damage to respiratory enzymes, for example, damage to cellular proteins, and also damage to other cellular components as well. Damage to the mitochondria can lead to dysfunctional mitochondria, and of course, because mitochondria are a major source of ATP, that can then limit generation of ATP. And one of the other targets for hydroxyradicals is DNA, both in the mitochondria and in the nucleus. And again, that can cause cellular dysfunction. There's some estimates about how much, there's no real estimates for how much hydroxyradicals are produced in a cell, but just to give you an idea, it's estimated that somewhere in the order of about 3,000 million hydrogen peroxide molecules are generated every hour in a single cell. So in other words, what you're looking at here are cells that are dealing with a substantial amount of reactive oxygen species production. So when we're discussing oxidative damage, you just need to be aware of some definitions. So when we're referring to oxidative damage, we're referring to the direct attack of reactive oxygen species during oxidative stress on macromolecules such as proteins, lipids, and DNA, and so on. The other point I would make is that normally these reactions are irreversible. So for example, if the protein is damaged by hydroxyradicals, and one or more of the amino acid residues are damaged, the only fate for proteins is breakdown into the individual amino acids and then removal of the damaged amino acids. So there's no real repair mechanism once the macromolecule has been targeted. With DNA, there can be excision of the damaged bases and replacement by a new nucleotide without damage. Now, in terms of how the cell is going to respond, there's at least 2 or 3 levels. So firstly, the cell can recover. So if the damage is minor, it can replace the damaged proteins, the damaged nucleotides, and continue on. a high level of oxidative stress, you can have persistent oxidative damage. That does normally affect some aspects of cell function, so you can consider the cell to be under stress. And then if the damage is severe enough, it can trigger cell death. It can trigger cell death by apoptosis, or if it's very severe, it will cause cell death by necrosis. Now, just to give you an idea of the targets within the cell, so this is some estimates, there's some modelling work that has looked at what hydroxyradicals would possibly attack in a cell, such as a leukocyte. And just again, remember these are estimates, but they give you a fair idea of where those hydroxyradicals are attacking. And you can see that over 2/3 of the macromolecules are likely to be proteins. In terms of RNA and DNA, you can see that is combined to about 13%, and you can see that attacks on lipids is considered to be very minor. My caveat on that is that if the hydroxyradicals are being generated within a lipid environment, you can expect lipids to be damaged. And then we've got damage on other types of macromolecules as well. This diagram summarizes the hydroxyradical damage concept. So firstly, we have to have generation of reactive oxygen species. That can be by an oxidative birth, for example, by immune cells, as a byproduct of cellular respiration, and we've seen some examples of that, and by a variety of environmental factors. The superoxide can then be dismutated into hydrogen peroxide, and then as a consequence of the Fenton reaction, we have the generation of hydroxyradicals. Depending on where the hydroxyradicals are generated, they can then go on to cause damage to proteins, to DNA, and to lipids. There has been a lot of work into looking at pathways by which hydroxyradicals causes damage to macromolecules. And it turns out that a lot of the degradation products can be used or developed into assays to track where hydroxyradicals are attacking. And we're going to look at 3 examples. One is protein damage, which is the carbonyl assay. Another is DNA damage, which generates a compound called 8-hydroxy-2-oxyguanosine. And the third example is lipid peroxidation, which generates isoprostanes. Now, I would say there are multiple other damage products as well, but we're going to focus on these 3 as our main ones to look at. So let's start with DNA damage. So estimates for damage to DNA by reactive oxygen species suggest something in the order of around about 100 hits per cell per day in healthy humans. So in other words, DNA is always being subjected to reactive oxygen species attack. What can happen to the DNA? You've got a variety of different attack sites, and each of these arrows, so if you're looking at this diagram here, we've got our ribose backbone here with our bases here, and each of these arrows represents evidence for where hydroxyradicals attacked. And really the point about this is that hydroxyradicals can basically attack anywhere. So that can lead to the scission of the deoxyribose ring. It can lead to base damage, so damaging these bases, and it can also lead to strand breaks as well. Now, what are the consequences to the cell? Well, you can end up with mutations. You can end up with translational errors. In other words, you're synthesizing dysfunctional proteins, and you can also inhibit protein synthesis as well. So damage to DNA is typically detected by measuring the products of the attack of hydroxyradicals on DNA. And so these degradation products can be measured by a variety of techniques, including high-performance liquid chromatography, gas chromatography linked to mass spectrometers, or HPLC linked to mass spectrometers as well. And there's also now a variety of antibody techniques that are available. However, I would say that there are ongoing arguments about which modification is best to measure, and also which particular techniques give you the most reliable results. So let's just pick one of the more popular products, so 8-hydroxy-2-deoxyguanosine, which is abbreviated as shown here. What happens with this base? It's an attack on the base. So we have guanine, we have hydroxyradical attack on the guanine, we have a number of rearrangements, and then we end up with this hydroxyl group on the guanine residue. Now, there is a major issue in measuring this compound, is that during the preparation of your sample, so you need to extract the DNA, you can end up with artifactual oxidation. And if that happens, then there is the potential to produce misleading results. So let me give you an example here. So one of the suggestions is that mitochondrial DNA is much more oxidized than nuclear DNA. The problem is that that's not necessarily true. There's ongoing controversy about it, and certainly some early studies by Halliwell indicated that that the amount of base damage that you saw in mitochondrial DNA could be less than in nuclear DNA. So when we're looking at these damaged products, we always have to keep in mind that there is still controversy about how reliable they are. Turning our attention to proteins, you saw on an earlier slide that proteins are probably where most of the oxidative damage is going to occur. So we can have oxidation of side groups, so the amino acid side groups of proteins. We can also have fragmentation of the polypeptide backbone, and we can also have cross-linking to other proteins. Now, there are multiple ways in which hydroxyradicals can damage proteins. The one that I would like to focus on is the one that leads to carbonyl groups, which can be detected. And this is one pathway, but not the only pathway by which carbonyls can be generated. So in this example here, you're seeing a hydroxyradical attack a carbon center, leaving us with a carbon radical. That carbon radical can then react with oxygen to form a peroxyl radical, and then that peroxyl radical can then form a carbonyl group. And that carbonyl group can be detected by a variety of different assays. Now, in terms of effect on function, it does depend on where the hydroxyradicals are attacking. So it is potentially possible for the protein to be damaged but continue functioning as normal. If it's an enzyme and the damage is in the active site, then usually you would expect that enzyme to become dysfunctional. When we're looking at the types of proteins, any type of protein can be basically affected by hydroxyradical attacks, so including things like receptors, enzyme, transport proteins, and so on. And you can also have secondary damage to other biomolecules. So, for example, inactivation of DNA repair enzymes can then lead to loss of fidelity in DNA polymerases in replicating DNA. So you can have a secondary consequence of this direct attack on proteins. So damage to proteins by a reactive oxygen species, the consequence really depends on the degree of damage. So you need a minimum level of damage before the protein is broken down by proteolysis. The mechanisms by which this happens are still not completely clear, but one possibility is that if there's increased hydrophobicity on the protein surface, that may render it more susceptible to protein degradation. And it can follow the usual process of protein degradation, which is through ubiquination, and then the degradation by the proteasomal process. Another pathway is by direct degradation by 20S proteasome. And that's shown on the right-hand side here in this diagram here. So we have our native protein. It's damaged. We have increased hydrophobicity on that protein, and the 20S proteasome can recognize that, so you do not require ubiquination. That leads to generation of peptides, and those peptides are then broken down into amino acids. The amino acids that are not damaged, which will be the majority of them, can be used to resynthesize new proteins, and those damaged amino acids can then be excreted from the body. So there are a number of techniques to detect oxidative damage on proteins. probably one of the most common ones that's used is the carbonyl assay. So that's the one that I'm going to discuss. The reaction mechanism is shown on the right hand side here. So here we have a protein with a carbonyl group reacting with 2,4-dinitryl phenylhydrazine to form this compound here, which absorbs very strongly at 360 nanometers. As a consequence, you can use spectroscopic methods to detect the presence of protein carbonyls. There's also now a variety of antibodies techniques that are available, which means you can set up ELISA assays or Western blot techniques to measure these proteins. Now, the technique's been used in tissue and in blood to detect protein carbonyls in plasma. And certainly you do see increases in protein oxidation associated with disease and also with aging. And the point of these observations is that they implicate oxidative stress in the pathogenesis of the disease, or in terms of exacerbating the disease severity both in disease and aging. Now, there is a bit of a caveat to 2,4-dinitryl phenylhydrazine because it will react with any carbonyl group. And so that means that carbonyls that arise as a result of protein glycation are also going to be detected by this technique as well. And so what that means is that an increase in protein carbonyls is not necessarily a direct result of reactive oxygen species, and you would need appropriate control experiments to test for that. Carbonyls can also be generated as a consequence of proteins reacting with aldehydes. Some of these can be formed during lipid peroxidation, so it's an indirect measure of reactive oxygen species. But we can also have direct oxidation of amino acid side chains, such as I showed you in one of the earlier slides, so oxidation of glutamate, for example. Let's turn our attention to lipids, particularly the phospholipids that you find in membranes. Now, the lipids in the membranes that are particularly susceptible to oxidative damage are the polyunsaturated fatty acids, or PUFAs for short. And these contain 2 or more diene bonds. So they're a major component of membrane phospholipids. They're there to allow membrane fluidity, so flexibility, and also they're important for structural integrity. And the point about this is that they're particularly susceptible to free radical attack. Now, the way this works is that with hydroxy radicals can abstract a hydrogen atom from the fatty acid. We can then have the formation of a peroxy radical, and that can then go on and cause the formation of further radicals. Now, this is shown on the right hand side here. So here's our fatty acid with multiple diene bonds. We have the abstraction of a radical by hydroxy radicals. This leads to an unstable carbon radical. We can have rearrangement of the structure, still a radical. It can then bind to oxygen to form a peroxy radical. Abstraction of a hydrogen atom, and we get the formation of a lipid peroxide. Now, what I would say about this is abstracting the hydrogen atom from another polyunsaturated fatty acid, so we've got this chain reaction of increasing generation of lipid peroxide. So if we go back to this system here, we've got the generation or the propagation of lipid hydroperoxides. So the point about this is one hydroxy radical doesn't lead to one lipid peroxide, it can lead to multiple lipid peroxides because we've got this ongoing chain reaction. So there's an amplification effect. So what are the consequences of lipid damage? Well, there are multiple potential consequences. And this diagram just gives you a bit of an overview of how hydroxy radicals can damage the phospholipid in the membrane. So we view this as the hydrophobic environment of a membrane. It could be the plasma membrane, it could be mitochondrial membrane. We have this hydrophobic component here made up of the polyunsaturated fatty acids, and we have the polar head interacting with the aqueous environment. So we've had a hydroxy radical attack on this polyunsaturated fatty acid here, we've had the formation of peroxy radical. Now the point about the peroxy radical is it's no longer hydrophobic, and so that means that we're going to have distortion of this phospholipid because the peroxy radical, which will form a lipid peroxide, will also be hydrophilic as well. Now because of this structural change, we're going to have changes in membrane fluidity. So membrane fluidity can decrease, so that means the membrane becomes stiffer. We can have an increase in membrane permeability, which will allow ions to cross the plasma membrane or the mitochondrial membrane, which will increase stress on the cell. If there are proteins within the membrane, that can affect membrane enzyme activity, for example, and also further breakdown of these phospholipids can generate reactive and quite toxic metabolites. Okay, so let's just turn our attention to what happens to the lipids once they're oxidized. So once the phospholipids have become oxidized, they are then degraded. And that means the phospholipid is basically destroyed and the fatty acid residues are released. And those fatty acid residues can then undergo a variety of degradative pathways. And some of those degradative pathways are shown here. So there's our polyunsaturated fatty acid, which will be part of a phospholipid. It's been damaged to form hydroperoxide. We've got release of the fatty acid residues, and then there's a variety of decomposition products. So we can form a variety of radicals, so alkoxy radicals, for example, and peroxy radicals. We have rearrangement of the structure. One of the ones that of particular interest to us is the isoprostanes. So these are formed from arachidonic acid. They're thought to have potential toxicity, but the reason these are particularly interesting to us is that they are also markers of lipid peroxidation. So they're quite widely used to track lipid peroxidation. There are a number of other decomposition products. Pentanes, for example, are volatile, can be released in the breath, and so therefore a few techniques have been developed to detect pentane. We can also form aldehydes. One of the most significant ones is malonaldehyde. So that has also been used and developed into an assay for lipid peroxidation. One of the other things I would say about this compound is it can also react with protein residues to form a protein carbonyl. And the last compound that's of interest to us is hydroxynonenal. It's thought to be a major end product, and it's also considered to be toxic to cells as well. And again, there are a number of assays that can be used to detect hydroxynonenal, or HNE for short. So, how do you detect damaged lipids? Generally speaking, the methods are always based around some degradation product of phospholipids. So, for example, assays have been developed to detect malonaldehyde. There are also an assay called the thiobarbiturate assay, acid reactive material assay, which is also abbreviated as TBARS assay. But the one that's considered to be the gold standard for detecting lipid damage is isoprostanes. So it's likely to be the best available biomarker of lipid peroxidation for human studies. And this just summarizes the process by which this happens. So here's our phospholipid here. We have lipid peroxidation. We end up with lipid hydroperoxide, which is non-enzymatic, by the way. We then have phospholipases that break down the phospholipids, and it produces a variety of different types of isoprostanes, and it's those isoprostanes that can then be detected as measures of this original hydroxy radical or carbon-mediated hydroxy radical attack. So in terms of measuring isoprostanes, there are a variety of techniques around. Certainly antibody techniques have become much more popular. But probably the gold standard is using gas chromatography. This involves a fairly arduous sample preparation step. putting the sample into gas chromatograph, where it separated out the compounds, and then running it through a mass spectrometer to pick up the particular isoprostanes that you're interested in. And generally these are the F2 alpha isoprostanes that we're interested in. In fact, one of the major papers for the development of this technique occurred in Royal Perth Hospital around about 20 years ago. So, let me return to this issue about detecting oxidative damage. Lipids, or detecting lipid peroxidation, is a good example of some of the difficulties we face when we're trying to work out whether oxidative damage has occurred in tissue. What I've highlighted here is 3 of the more common techniques. So F2 isoprostanes, malonaldehyde, and the T-bars assay. And so the question we're asking is, in vivo, so in an animal model or in humans, are these compounds good biomarkers of lipid peroxidation? In other words, are they telling us the degree to which lipid peroxidation is occurring? So let's start with the F2 isoprostanes. They're specific end products of the peroxidation of polyunsaturated fatty acid. That's good. That means that you're not going to be confounded by other potential interfering compounds. So, for example, we take protein carbonyls. Protein carbonyls can occur as a result of glycation. This is not going to happen with the isoprostanes. A second potential issue is diet. Are there oxidation products in food that can affect your interpretation? So the good thing about isoprostanes is that's not a problem. Another potential problem is storage. You're collecting these compounds. If their composition changes during storage, then you're not going to get an accurate reflection of what's happening in the tissue. So again, this is not an issue with the isoprostanes. And we also have very good and accurate methods for measuring these isoprostanes. So overall, it looks like isoprostanes are quite a good way to monitor lipid peroxidation. Now, I would have a bit of a caveat on this, is that when we're looking at humans, most of the time the isoprostanes are measured in plasma, extracted from blood, or in urine. So these provide a whole body measurement, and it's not necessarily clear where the isoprostanes are being generated in the body. But as I indicated before, isoprostanes are considered to be the gold standard for measuring lipid peroxidation. However, preparation of the sample for measurement is technically fairly complex, and you're also using equipment that's quite expensive and may be difficult to access. And so as a consequence, a multiple of other methods are used. A common one, for example, is malonaldehyde, or MDA for short. But the problem with malonaldehyde is that you can find malonaldehyde in diet. And so that means that changes in malonaldehyde, for example, in urine, could be consequences of changes in diet. So in other words, you end up being misled. An extremely popular assay for lipid peroxidation is called the T-bars assay. It's very popular because it's very cheap, and the equipment that you need to use is found in a wide variety of laboratories. So, for example, UV plate spectrometers, which you've already been exposed to or know something about. And with this reaction, we have thiobarbiturate acid reacting with hydroperoxide to form a fluorescent red product. But it's unlikely this assay is actually any good, because most of the TBA reactant material you find in body fluids is actually not related to lipid peroxidation. And yet this is a very, very popular technique for measuring apparent lipid peroxidation. So you can see that there is that potential that many people will be misleading themselves. So I have been using this term biomarkers. So let me explain what a biomarker is. A biomarker is some indication that oxidative event has occurred. So for example, we're looking at oxidative stress. The presence of that biomarker or fingerprint can be used to infer that reactive oxygen species have been formed. And I've given you some example, protein carbonyls, for example, isoprostanes, malonaldehyde, and so on. So if they go up in whatever tissue that you're measuring, then the inference is that you've had an increase in reactive oxygen species production. We can use biomarkers in a variety of ways. One way is to use it to check whether antioxidant therapies work. So if you think that oxidative stress is exacerbating a disease or causing a disease or a condition, you can try treating it with synthetic antioxidants or dietary supplements, which are antioxidants, to see if you can block the oxidative damage and, as a consequence, improve outcomes. Now, biomarkers are used in animal studies. We've certainly used them ourselves, but generally speaking, you find wide use of biomarkers in humans. And that's because with humans, generally speaking, you can access urine samples and blood samples, much more difficult to acquire tissue samples, so muscle samples or liver samples, for example. And the way that we develop and use biomarkers is shown on the right-hand side here. So firstly, you're looking at people who have oxidative stress associated disease. This might be early in the disease, it might be late in the disease. And so what you're then interested in is identifying biomarkers that can track that disease, perhaps in the early stage or the late stages of the disease. On the right-hand side here, we've got a variety of different types of biomarkers that are potentially used or are useful. So, for example, I've discussed some of these. Isoprostanes, you'll see here, malonaldehyde, hydroxynonenal, and for a variety of other compounds as well. So, you then look for these biomarkers in the blood or the urine that you're interested in. Generally speaking, it's not advised to use a single biomarker these days because the potential to be misleading. So, for example, if the oxidative stress event was affecting primarily proteins, it wouldn't be much good measuring isoprostanes because you'd miss the oxidative stress event. So generally, the idea is to measure multiple biomarkers, or that's one of the suggestions, to get an indication of the health status of the subject. You then give your subjects the particular antioxidants that you're interested in. That antioxidant may be effective. So in other words, you've got an effective antioxidant therapy. The antioxidant therapy might rescue and prevent damage to macromolecules in cells, but dosing then becomes an issue, and if there's not sufficient dosing, then you're going to end up with an ineffective antioxidant therapy. But generally speaking, what you're doing is tracking the potential of antioxidants to affect these biomarkers. So the idea would be that if isoprostanes started coming down once you treated with your antioxidant, then you're likely to have an effective antioxidant treatment. So this is an example of using isoprostanes as a biomarker of oxidative stress. In this particular study that you're looking at, which was published some time ago, what they were looking at is whether you could use polyunsaturated fatty acids, so eicosapentaenoic acid or dodecahexaenoic acid, as antioxidants. In other words, did they decrease oxidative stress, and did they decrease inflammation in hypertensive type 2 diabetes? And what you're looking at here is the results of that study. There was an olive oil control group, and then we had our two polyunsaturated fatty acids, EPA and DHA. And what they've done is measured it relative, so they've measured it pre and post treatment. So in our control, we've had no significant changes in our isoprostane levels. With EPA, we've had a significant decrease in our EPA level, in our isoprostane levels, and with DHA, we've also had a significant decrease in our isoprostane levels. So what this is suggesting is that both of these compounds are decreasing isoprostane, so in other words, are likely to decrease oxidative stress in the body. Whether it's sufficient to have a clinical beneficial outcome would require additional measures. But this does give you an idea of how you use these biomarkers. The last point I want to make in the context of using biomarkers is when you're asking questions about the type of oxidative stress you have in humans or in animal models. And the recommendation is to use more than one biomarker. Let me give you an example. We work on muscular dystrophy. One of the questions that you might have about muscular dystrophy is there oxidative stress in muscle? And the reason we're interested in oxidative stress in the muscle is because muscular dystrophy is a wasting disease of the muscle, which is eventually fatal for the boys that are involved. And so what we're interested in is whether oxidative stress is contributing to that muscle wasting. To do that, we need to identify the type of oxidative stress we're dealing with. Now, we've looked at two biomarkers of oxidative stress. The first one is the protein carbonyl assay. This just shows you some ways in which those carbonyls can be formed. These are metal catalyzed reactions, so we have proline here, side group, arginine and lysine. All of these can form protein carbonyls, which we can then measure. We also looked at malonylaldehyde. Because we controlled the diet, we were confident that we weren't getting artifact because of the diet. And just let me remind you that the malonylaldehyde comes from lipid peroxidation. So we've got our polyunsaturated fatty acids being attacked by hydroxyl radicals, formation of a carbon radical, reaction with oxygen, chain reactions producing more lipid radicals. But we eventually get a breakdown of these lipid hydroperoxides to form malonylaldehyde, and we also measured that. So what we did is we looked in the muscle of a muscular dystrophy animal model, which was the MDX mouse, to see whether malonylaldehyde or carbonyls were affected by the disease. So if we look at malonylaldehyde, for example, you can see that there's no significant difference between our MDX or muscular dystrophy model and our control model. On the other hand, if you look at the protein carbonyls, you can see that the MDX are showing significantly higher levels of protein carbonyls than the controls. So what this is telling us is that it's likely that the pathology associated with dystrophy, if oxidative stress is involved, is going to be through damage to proteins, with lipid peroxidation likely not to be as important. But really the point about this is that if you'd gone in and you just measure malonylaldehyde, you would have missed the importance of the other damage pathway, which is through proteins. So this is why you've got this recommendation to use multiple biomarkers. And it also gives you an idea of some of the complexity that's associated with this type of research. Okay, so that completes the discussion about oxidative damage. So just a few points here that you need to be able to understand and explain in terms of reactive oxygen species, how they can damage protein, lipids and DNA, and also how we can use that information in terms of generating analytic assays to monitor the different pathways for oxidative damage. And then the last part of this lecture was really about biomarkers, so you need to understand something about biomarkers. A couple of key references that are worth looking at is this one by Winterborn, looking at the biology of reactive oxygen species, and this older one by Halliwell and Whiteman, talking about how to measure reactive oxygen species and oxidative damage in vivo and also in cell culture. This paper in particular highlights some of the issues that we're faced with when we're trying to look at oxidative stress in tissue. And both of these publications are on the LMS site.